For most of human history, the idea of finding an animal from the Ice Age perfectly preserved, fur soft, organs intact, blood still liquid, belonged to the realm of legend. The ancient world seemed separated from the present by a barrier impossible to cross. Bones and fossils were the closest anyone could get to animals that vanished tens of thousands of years ago. But the permafrost of the northern hemisphere began revealing something no fossil bed had ever offered: complete bodies, frozen with such precision that time barely touched them. But before we jump back in, drop a comment letting us know what time you're tuning in and where you're watching from. We love seeing how far our episodes are reaching. And don't forget to hit that subscribe button. It really helps us keep bringing exciting topics here on the Primal Roots channel. The discovery of these creatures transformed scientific understanding of extinction, climate, and evolution. They emerged from the melting ground as if waking from a long sleep, carrying within their tissues stories locked away since mammoths roamed alongside early humans. Each thawed carcass became a biological time capsule, opening a window into ecosystems that had vanished long before civilization began. The ice had not merely preserved shape or structure, it had safeguarded life itself at the molecular level. One of the most extraordinary revelations came in 2010, when researchers working in Siberia uncovered the remains of a young woolly mammoth. Nicknamed Yucca, the juvenile female lay exactly where the ice had held her for nearly 40,000 years. Her fur retained color, her skin remained supple, and fat tissue still clung to her frame. What shocked the excavation team was the presence of liquid blood trapped inside her body. Hemoglobin flowed from the carcass when they attempted to move her, behaving not like ancient residue, but like blood from a recently deceased animal. This phenomenon challenged previous assumptions about biological limits. Blood should have crystallized under such low temperatures, yet yuccas remained fluid. Scientists proposed that mammoths possessed natural cryoprotectants, biological compounds that lowered the freezing point of bodily fluids. If true, it meant that Ice Age giants had evolved biochemical defenses, similar to those found in certain Arctic fish and amphibians today. Yucca was not the only frozen mammoth discovered, but her preservation represented a milestone. Tissue samples allowed researchers to examine muscle fibers, cellular structures, and even the arrangement of nerve bundles. Under the microscope, her biology looked impossibly fresh. This level of preservation opened new opportunities for genetic research, pushing the long-standing questions of de-extinction from science fiction into possible future reality. Not far from where Yucca was found, a smaller frozen creature captured global attention: a 40,000-year-old wolf, discovered intact with fur, teeth, and facial features preserved with extraordinary clarity. The animal's muzzle, ears, and whiskers remained undamaged, appearing almost animated beneath the frost. Unlike modern wolves, its skull carried unique proportions, suggesting a lineage distinct from those alive today. DNA analysis later confirmed that this wolf belonged to an early branch of canids that bridged ancient wolves and the first domesticated dogs. The permafrost had protected not only its body, but an evolutionary chapter that had vanished everywhere else. The discovery deepened understanding of the relationship between early humans and predatory species. The remains of this wolf, combined with genetic links to early domesticated dogs, illustrated a landscape where large predators overlapped with human populations in ways both dangerous and essential. Humans may have competed with these wolves for food, while also benefiting from observing their hunting patterns, their territorial movements, and eventually forming symbiotic relationships that shaped the development of early domesticated animals. Perhaps the most striking frozen findings came from Pleistocene cave systems that periodically collapsed or became sealed by ice, trapping animals inside. In several cases, researchers found the bodies of extinct cave lions, preserved with such detail that the texture of their whiskers remained visible. Their golden fur still clung to their bodies, and their claws showed no signs of decomposition. These cats were apex predators of their time, moving silently through forests and grasslands where early Homo species built shelters, hunted, and struggled for survival. Their perfectly preserved remains provide a tangible encounter with a creature that had once dominated the same terrain as our ancestors. Some of the most fragile creatures found in permafrost are surprisingly well-preserved infants, mammoth calves, baby cave lions, and even a rhinoceros calf from more than 34,000 years ago. These small bodies endured the freezing process with astonishing completeness. In some cases, their eyelashes, hooves, and internal organs remained unaltered. Their vulnerability in life became their advantage in death. Young animals often died quickly from environmental stress, predators, or accidents, freezing before decomposition could begin. These discoveries revealed a more intimate side of Ice Age ecosystems. They showed herd structures, growth patterns, and survival challenges faced by the young of large species. The ability to analyze milk residue, stomach contents, and bone marrow offered unprecedented insight into how these animals lived and died. The frozen rhinoceros calf, for example, contained partially digested vegetation that identified the seasons in which it fed and the plant communities that surrounded it, plants that no longer exist in the modern Arctic. As paleontologists worked to understand the frozen specimens, another layer of significance emerged: the human connection. These animals were not isolated beings living in remote Pleistocene landscapes. Many walked the same paths as early human groups migrating across Eurasia. Archaeological evidence suggests repeated encounters, both cooperative and adversarial, between humans and the megafauna of the Ice Age. Some animals became symbols in early art, etched into cave walls by hands attempting to understand or honor the creatures that shaped their world. Others became essential sources of food, clothing, tools, and spiritual meaning. Frozen remains preserve traces of these interactions. Mammoth bones carried cut marks from stone tools. Cave lion hides showed evidence of skinning. In rare cases, spear tips were found near the bodies. The ice served as a witness to this shared existence, freezing moments that linked humans and animals across thousands of generations. Even the odors preserved in frozen carcasses stunned researchers. When permafrost thaws, the smell of ancient organic matter rises from the body, an earthy, slightly sweet scent unlike any modern decomposition. These odors reveal that organic compounds in tissues survived intact, giving scientists clues about diet, habitat, and even the bacteria present at the time of death. In exceptionally preserved specimens, researchers detected lipid residues and proteins that helped reconstruct the physiology of species long extinct. Some discoveries offered unsettling implications. In certain carcasses, researchers found preserved parasites, bacteria, and viral fragments. Though no active pathogens have yet revived intact from these animals, the fact that microbial structures survived in frozen tissue underscored the risks associated with accelerating permafrost thaw. Ancient microorganisms, dormant for tens of thousands of years, may resurface as global temperatures rise. Yet beyond these risks, the frozen creatures also offered extraordinary scientific opportunities. They presented a level of biological detail never before available for extinct species. For the first time, scientists could examine muscle fiber arrangement, blood vessel structure, stomach bacteria, and genetic sequences extracted not from mineralized bone but directly from preserved tissue. Each discovery deepened understanding of Pleistocene ecosystems, revealing complex interactions that shaped evolutionary paths leading to modern species. The presence of blood in some specimens sparked discussions about de-extinction technologies. If intact DNA fragments could be extracted from blood, muscle, or organ tissue, then cloning or hybridization efforts might one day revive aspects of these species. Although complete cloning remains impossible due to DNA degradation, the mere possibility created intense scientific debate. Could the essence of a species return through genetic engineering? Should humanity attempt to resurrect extinct creatures? And if revived, what role would they play in modern ecosystems? These questions began taking shape as laboratories worldwide started sequencing DNA from increasingly well-preserved specimens. Researchers reconstructed large portions of mammoth genomes, identified traits useful for cold resistance, and explored ways to combine these traits with modern elephant DNA. The frozen animals were no longer inert relics. They were genetic archives capable of influencing the future. As more discoveries emerged from melting ice, the scope of the frozen world expanded. The permafrost revealed steppe bison, musk oxen, horses, foxes, and birds from ages long past. Some creatures showed signs of sudden death, perhaps from storms, predators, or environmental events. Others appeared to have died peacefully, lying down as the cold overtook them. Every body added new information about the biodiversity of a world lost to time. The unfolding of these discoveries marked a turning point in understanding the Ice Age. The frozen ground became a library of vanished ecosystems, revealing life with a clarity that no fossil, cave painting, or myth could match. These preserved creatures demonstrated that extinction does not always erase. Sometimes it stores, seals, and waits. With each body recovered from the ice, the boundary between the ancient and the modern blurred. The past no longer felt distant. It felt close, vivid, and almost breathing, held in the cold earth until the world became warm enough to unearth the memories it had been keeping. Long before frozen mammoths were unearthed from the Arctic permafrost, their towering silhouettes shaped the daily lives of ancient humans. These enormous animals roamed the same valleys and plains, leaving deep footprints in the soil that early human groups crossed during hunts, migrations, or desperate searches for shelter. The creatures whose bodies now emerge from the ice were once the dominant figures of landscapes humans struggled to understand, navigate, and survive. Their presence influenced the development of early behaviors, carved pathways through continents, and left traces in the earliest symbols ever created. Each time a frozen mammoth or cave lion is uncovered today, it revives a chapter of this shared history. These animals were not distant from human life. They served as threats, resources, spiritual icons, and reminders of the fragile balance between human survival and the overwhelming power of nature. Their remains in the ice reflect intense climatic conditions, but they also echo the long coexistence between our species and these megafauna giants. Archaeological sites across Eurasia reveal that humans encountered mammoths with a mixture of fear and opportunism. Mammoth bones bearing cut marks indicate that humans scavenged or hunted these giants whenever conditions allowed. Their meat sustained communities through harsh winters, their fat provided vital calories, and their hides became shields against sub-zero temperatures. A single mammoth could support an entire group for weeks, offering nourishment at a time when plant resources were scarce and competition with predators was constant. The frozen remains uncovered today show details that early humans witnessed with their own eyes. The thick layers of hair, the large domed heads, the tusks twisting like ivory spirals. These features were familiar to ancient people. Some caves in Europe contain paintings of mammoths that capture musculature, posture, and movement with remarkable accuracy. These depictions were not mere ornamentation. They were expressions of observation, memory, and communication. Every line drawn in ochre reflects a moment where human life intersected with the formidable presence of these animals. Other frozen creatures tell similar stories. The intact cave lions found in Siberian ice recall a world where predators dominated ecosystems, and humans had to constantly adapt to avoid becoming prey. Cave lions, with their muscular bodies and keen hunting skills, played a significant role in the dynamics of ancient survival. People living during the Ice Age recognized their strength, their stealth, and their dominance. The preserved remains show pelts thick enough to withstand brutal winters and claws suited for capturing large herbivores. Humans learned to read their tracks, avoid their dens, and adapt strategies to protect themselves from ambush. These interactions between humans and predators shaped human behavior. Survival demanded cooperation, tool innovation, and constant situational awareness. The very act of moving across the landscape required a heightened understanding of animal movement, seasonal patterns, and the dangers lurking in every terrain. The fact that early humans survived side by side with creatures like cave lions is a testament to the ingenuity that allowed humanity to expand across continents. The frozen wolf, discovered with its face preserved in extraordinary detail, provides another link to early human life. Wolves were not only competitors, They were also early partners. The preserved animal belonged to a lineage bridging ancestral wolves and the first domesticated dogs. Its frozen features, sharp muzzle, dense fur, strong jaw, mirror the wildness of its era. Yet genetic studies indicate that some wolves eventually developed mutualistic relationships with humans. Shared hunting grounds brought opportunities for interaction, and over generations, wolves evolved alongside people into the first domesticated dogs. The frozen specimen stands not just as a relic, but as a marker of the earliest steps toward one of the longest-standing animal-human partnerships in history. These creatures shaped human culture as much as they shaped survival. The sacred symbolism of mammoths appears in carvings and figurines, representing strength, endurance, and reverence for the natural world. Lions symbolized courage and danger, while wolves became icons of intelligence and loyalty. These animals were perceived not only as prey or predators, but as forces woven into the identity of early societies. Their frozen bodies, now emerging from the permafrost, reflect a time when humans expressed their relationship with nature through art, rituals, and oral traditions. Some of the bodies recovered from ice show signs, suggesting human involvement. Mammoth remains occasionally contain embedded spear points or evidence of coordinated hunting activity. These marks indicate moments when human groups challenged the dominance of giants, risking their lives to secure enough resources to sustain their communities. The frozen remains illustrate the brutality and ingenuity of these encounters. A single miscalculated strike could cost a life, yet humans persisted, adapting techniques that allowed them to exploit opportunities that predators often left behind. The environment that fostered these interactions was unforgiving. Glaciers advanced and retreated, shaping rivers, freezing valleys, and altering the availability of resources. Early humans had to learn how to endure seasons marked by long nights, freezing winds, and landscapes devoid of edible plants. The presence of large animals offered both danger and hope. A mammoth migrating through the snow was a threat to be avoided, or a prize to be pursued. Success or failure in approaching such a creature could determine whether a community survived the winter. Frozen discoveries also reveal a deeper, more emotional aspect of life in the Ice Age. Among the preserved creatures are infants, mammoth calves, lion cubs, and other young animals that met tragic ends in the cold. Their bodies froze rapidly, with fine details preserved: eyelashes, whiskers, small hooves, and delicate skin. These remains highlight the vulnerability of life during the Pleistocene. A sudden blizzard, a misstep near a frozen river, or separation from a mother could result in instant death. Early humans encountering these frozen bodies may have recognized the fragility mirrored in their own young. Survival during this era required constant vigilance and a deep understanding of nature's unpredictability. Humans also made use of the permafrost without realizing it. The Earth served as a natural freezer long before humans created storage techniques. During periods of scarcity, carcasses buried in snow or frozen ground became valuable reserves. Observations of frozen animals may have taught early groups how to preserve meat for extended periods. In this sense, the frozen creatures of today reflect ancient strategies of resource management that were lost as climates warmed and societies evolved. The connection between humans and these Ice Age animals extended beyond physical interaction. The animals influenced migration routes, settlement patterns, and cultural practices. Herd movements dictated where humans traveled, while predators shaped where shelters were built. In some regions, humans positioned themselves along migration corridors to intercept large herbivores at predictable times of year. The frozen specimens discovered today map these ancient patterns, revealing not only the biology of the animals, but the geography of human survival. Some frozen remains contain plant matter and stomachs, revealing what the animals ate in their final hours. This information reconstructs Ice Age ecosystems with remarkable accuracy, identifying plants that humans also consumed or used as materials. The digestive contents become evidence of shared habitats and overlapping resources. The diets of these creatures tell stories of seasons, climate variability, and the structure of ancient landscapes. The profound stillness preserved in these bodies contrasts sharply with the constant motion that defined their lives. The mammoth trudging across snowy plains, the lion hunting in twilight, the wolf tracking scents through dense forests. All of these moments ended in sudden cold, locking the animals into a timeless state. For humans living in the same era, these animals were part of a world that demanded resilience. Their frozen forms bring that world back with clarity that words alone cannot achieve. As these creatures rediscovered daylight after tens of thousands of years, they reawaken an ancient relationship between humans and nature. They remind modern audiences of a past where survival required confronting predators larger than vehicles, navigating landscapes shaped by ice, and interpreting the behavior of animals that dominated ecosystems long before cities existed. The frozen bodies represent more than scientific data. They represent chapters of human understanding, fear, reverence, and adaptation. Each discovery makes the Ice Age feel less like a distant epoch and more like a previous chapter of a story still unfolding. Humans today inherit landscapes altered by climate and time, yet the frozen remnants of mammoths and predators preserve moments when humans and megafauna shared the same ground. These encounters shaped human evolution, influencing tools, strategies, art, and early cultural expression. The reappearance of these creatures in modern times invites reflection on how deeply intertwined human history is with the natural world. Their presence reveals how early humans learned, adapted, and endured. It shows that humanity did not rise in isolation, but rather in dialogue with the animals that surrounded it, sometimes adversaries, sometimes resources, and often symbols that shaped early imagination. The discovery of entire Ice Age creatures preserved in permafrost did more than reveal their physical forms. It exposed cellular landscapes that once pulsed with life, muscles that remained elastic, organs holding original structure, and blood that resisted crystallization opened a scientific frontier once believed impossible. Frozen bodies offered molecular information with a level of resolution unmatched by traditional fossils. From this point forward, the study of extinction no longer focused solely on bones. It shifted toward the chemistry of life itself, leading to one of the most ambitious scientific pursuits of the century: the attempt to revive vanished species. Genetic material found within frozen specimens opened the door to reconstructing the genomes of extinct animals. DNA fragments, though damaged by time, retained enough information to map large portions of genetic sequences. The first successful extraction of high-quality mammoth DNA astonished geneticists. Large regions of their genome revealed adaptations for cold resistance, fat storage, oxygen transport, and hair density, traits that made mammoths extraordinary survivors of the Pleistocene. With each sequencing project, the idea of restoring such species moved from speculation to structural possibility. The renewed interest in de-extinction intensified when scientists examined preserved soft tissue under high-resolution microscopes. Some cells retained recognizable membranes, partially intact nuclei, and chemical traces of their original biochemical functions. These structures represented a biological blueprint frozen in time. Though the DNA inside them had fragmented over millennia, its presence was enough to allow advanced sequencing technologies to reconstruct large portions of the genome through comparison with modern relatives. Among the most significant breakthroughs were molecular findings within mammoth muscle tissue. Researchers detected proteins and lipid profiles that did not merely resemble those of their modern cousins, but revealed how these giants processed energy in harsh environments. Fat deposits within frozen tissue showed chemical signatures adapted for extreme cold. Traces of brown adipose tissue indicated high thermogenic capacity. These insights helped explain the mammoth's survival strategies, but also provided potential guidelines for future genetic engineering projects. Cloning, however, requires more than DNA. It demands intact nuclei capable of directing embryonic development, structures that time rarely spares. Despite impressive preservation, ancient nuclei deteriorate, fragment, and lose the integrity needed for traditional cloning methods. Attempts to extract whole, viable nuclei from frozen mammoth cells consistently failed. While the cell walls and shapes were often preserved, internal components collapsed or degraded beyond functional recovery. Recognizing these limitations, scientists turned to alternative approaches. Instead of cloning in the classical sense, they began exploring genome editing techniques that could transfer mammoth genetic characteristics into the cells of modern elephants. The Asian elephant, sharing a close evolutionary relationship with mammoths, became the primary candidate for hybridization efforts. Researchers focused on identifying mammoth genes responsible for cold adaptation, genes involved in hair growth, fat metabolism, oxygen binding, and temperature regulation. Using gene editing tools, these sequences were inserted into elephant cells, creating preliminary hybrids at the cellular level. These modified cells demonstrated early signs of functional expression. Mammoth hair genes activated in controlled laboratory environments. Cold-adapted hemoglobin sequences showed expected oxygen-binding behaviors. Lipid metabolism exhibited the patterns associated with Ice Age survival. These experiments did not produce living animals, but they represented meaningful progress. The genetic signature of mammoths was no longer confined to frozen tissue. It appeared in living cellular systems for the first time in thousands of years. The discovery of liquid mammoth blood intensified the debate around de-extinction. When researchers moved yucca, the juvenile mammoth preserved in Siberian ice, they witnessed dark red fluid emerging from her body. The implications extended far beyond sensational headlines. The presence of hemoglobin with reduced freezing point suggested an evolutionary adaptation similar to antifreeze proteins found in certain Arctic fish. If this adaptation existed, it meant that mammoths possessed molecular strategies to survive drastic temperature fluctuations. The existence of preserved blood also allowed researchers to analyze heme structures and protein stability with unprecedented clarity. Scientists engaged in long-term efforts to recreate mammoth hemoglobin in laboratory environments, comparing ancient proteins to those of modern elephants. The differences revealed how mammoths delivered oxygen efficiently in sub-zero climates. Such findings continued to fuel discussions about the potential viability of genetically engineered mammoths or mammoth-like hybrids. Specimens of frozen cave lions, wolves, and other predators also contributed to genetic investigation. Cave lion cubs preserved with remarkable fidelity allowed the recovery of DNA fragments strong enough to map portions of their genomes. These sequences helped identify distinctions between cave lions and modern lion lineages. Analyses revealed adaptations for colder climates and hunting strategies suited for open tundra environments. Their genetic legacy represented an apex predator that shaped the ecological balance of the Pleistocene. The frozen wolf head found in Siberia presented unique opportunities. The animal belonged to a lineage bridging early wolves and the ancestors of domestic dogs. DNA extracted from its tissue helped clarify the evolutionary timeline of canids, offering insights into when and how domestication pathways diverged. Such discoveries not only reconstructed ancient species, but also illuminated the origins of one of the most significant human-animal relationships in history. Yet even with these successes, the technical challenges of de-extinction remained formidable. DNA recovered from frozen specimens appears in fragmented pieces, often damaged by oxidation, radiation, and time. To reconstruct genomes, scientists must align these fragments with the DNA of modern relatives, identifying overlaps and inferring missing regions. This method produces a comprehensive sequence but not a perfect replica. Over thousands of years, some genetic information becomes irretrievably lost. Consequently, any revived species would be a close approximation rather than an exact copy of its ancient counterpart. The issue of surrogate mothers posed additional obstacles. If hybrid embryos were created, they would require gestation within a modern elephant. Elephants, however, are endangered and have long gestation periods, making experimental reproduction both risky and ethically complex. Artificial womb technologies, though advancing quickly, remain far from ready to support the development of large mammals. These limitations forced researchers to balance ambition with caution. Some frozen specimens contained more than muscle and bone. Trace amounts of stomach content preserved details about diet, microbiome composition, and environmental exposure. These biological remnants helped reconstruct Ice Age biodiversity and informed what revived or hybridized species might require to survive. A revived mammoth could not simply exist in the modern world without access to the plants and landscapes that sustained its ancestors. Studies of preserved stomach matter identified grasses, herbs, mosses, and shrubs long extinct or dramatically changed. Such findings raised questions about whether de-extinction projects should also consider ecological reconstruction. Additional breakthroughs came from studies of preserved soft tissue microbiomes. Frozen stomach bacteria, though no longer active, retained structural signatures revealing the digestive ecology of Ice Age species. Recreating such microbiomes in laboratory environments might one day support the health of genetically engineered hybrids. But replicating complete ancient digestive systems remains a distant goal, requiring the interplay of genetics, microbiology, and ecosystem management. Alongside these biological insights, frozen remains exposed the extraordinary variety of animal life that shared the earth with early humans. Musk oxen with pristine fur, horses with intact manes, bison with well-preserved horns, and even birds with feathers still attached, all emerged from thawing soils. These animals formed dynamic communities that shaped ecosystems across the northern hemisphere. Their genetic material, though difficult to fully reconstruct, provides a clearer picture of the lost biodiversity that once defined glacial landscapes. Researchers examining frozen tissue uncovered more than evolutionary secrets. They discovered molecular remnants that allowed them to model ancient physiology with surprising precision. Mitochondrial DNA sequences traced maternal lineages across continents. Collagen peptide profiles mapped evolutionary divergence among ungulates. Amino acid ratios revealed nutritional stress and environmental conditions surrounding the animal's final days. Each molecular clue contributed to a broader understanding of how Ice Age mammals adapted to environments frequently reshaped by cold, scarcity, and shifting terrain. The field of synthetic biology expanded further when scientists succeeded in reconstructing proteins from extinct animals using fragments decoded from frozen remains. These experiments produced functional proteins in laboratory settings, echoing behaviors of their ancient counterparts. While such achievements did not recreate full organisms, they demonstrated the feasibility of reviving biological components of long-lost species, a precursor to any larger revival effort. As technological capacity increased, debates intensified. Some researchers envisioned giant Ice Age herbivores filling ecological roles that had since disappeared, helping restore grasslands and reshape permafrost regions. Others warned about the unpredictable consequences of introducing genetically engineered species into modern ecosystems. These discussions influenced guidelines shaping how far research initiatives should extend into de-extinction efforts. Despite obstacles, the work continued to advance. Every newly thawed specimen provided additional fragments of ancient genomes, expanding libraries of genetic information. Different species offered varying levels of preservation. Mammoths and woolly rhinoceroses often yielded richer tissue samples, while cave lions and wolves contributed insight into predatory lineages. Collectively, these discoveries supported the largest genetic reconstruction of extinct megafauna ever attempted. Frozen bodies recovered from permafrost reshaped technological development. Cryogenics, genetic mapping, and genome editing all accelerated in parallel with Ice Age research. The ability to study ancient biological systems at such depth encouraged the development of techniques capable of extracting and interpreting smaller, more degraded DNA fragments than ever before. This technology benefited not only paleogenetics, but also modern conservation efforts, forensic science, and medical research. The challenges of de-extinction affirmed the importance of maintaining the balance between ambition and feasibility. Ancient DNA research continued uncovering details of life that seemed impossible to access a century earlier. Frozen creatures preserved not only their physical forms, but the chemical instructions that governed them. Their molecular echoes fueled questions previously unimaginable and pushed genetics into a new era defined by precision, adaptation, and advanced reconstruction. The ice did not merely store bodies. It protected blueprints of life written in fragile chemical code. The frozen tissue of mammoths, lions, wolves, and other creatures provided source material for understanding ancient ecosystems and exploring the limits of genetic engineering. The boundaries between past and future narrowed in laboratories where DNA fragments once locked in permafrost became active components of modern scientific inquiry. Permafrost has always acted as one of Earth's coldest vaults, sealing away organic matter, preserving entire ecosystems, and halting the natural decay that transforms life into soil. Everything trapped beneath its surface, plants, animals, microbes, spores, became suspended in a state between life and death. For tens of thousands of years, this frozen layer served as both a barrier and a reservoir, containing biological material from eras predating human civilization. As global temperatures rise and permafrost thaws at unprecedented rates, this vault is opening, releasing remnants of ancient worlds into modern environments. The frozen bodies of mammoths, cave lions, wolves, and other megafauna drew global attention, but those large creatures are only a fraction of what the permafrost contains. Much smaller and far more enigmatic organisms lie beneath the surface. Microbes, viruses, parasites, and fungi endured freezing cycles that would have destroyed most modern pathogens. Their survival reveals biological resilience that challenges assumptions about microbial dormancy. Some of these organisms may be inactive, others merely dormant, and a few potentially viable under the right conditions. Researchers studying permafrost samples discovered that microbial communities could remain preserved for tens of thousands of years. In experimental settings, certain bacteria resumed metabolic activity upon thawing. Their reanimation demonstrated that life can endure extreme cold with unexpected tenacity. Microbial spores, protected by thick walls, survived conditions that sterilize modern environments. These resilient microbes offer insight into ancient ecosystems, but they also present challenges regarding how thawing soils may reshape contemporary biological landscapes. Ancient viruses have also been recovered from deep permafrost layers. These viruses, often termed giant viruses due to their unusual size, contain genetic material capable of infecting single-celled organisms. In controlled laboratory conditions, some viruses revived after thawing, successfully infecting hosts specifically chosen to prevent any risk to humans or animals. Their revival confirmed that genetic structures can survive deep time, frozen but intact. The existence of viable viruses in permafrost introduces questions that extend beyond academic curiosity. Although no known viruses recovered so far pose threats to modern humans, the potential for unknown viral forms to emerge increases as deeper layers thaw. These viruses may have interacted with Ice Age mammals, early species of megafauna, or ancient microbial ecosystems. Their characteristics remain largely unexplored. Because viruses evolve rapidly, ancient strains could differ dramatically from any found today. Understanding their behaviors requires systematic research to prevent unexpected consequences as the Arctic continues warming. The release of ancient microbes and viruses coincides with shifts in ecosystems that once relied on stable frozen ground. Permafrost acted as a structural foundation for vast stretches of tundra. As thawing accelerates, landscapes collapse, releasing trapped greenhouse gases and exposing newly uncovered organic layers to the air. These changes alter microbial activity in the soil, triggering cascading chemical reactions. Once frozen carbon begins decomposing, producing gases that further accelerate warming. The biological processes interacting within these thawing soils form a feedback loop that scientists continue to study. In addition to microbes, thawing permafrost reveals the remains of parasites that once inhabited Ice Age animals. These include insect larvae, intestinal parasites, and protozoa preserved in tissue samples. Their structural integrity reflects conditions that could allow partial reactivation under ideal circumstances. While most parasites depend on living hosts and are unlikely to resume their life cycles, their preservation still provides valuable insight into the health challenges that ancient animals faced. Studying these preserved parasites helps reconstruct broader ecological networks, revealing predator-prey dynamics and patterns of disease transmission in prehistoric times. Frozen ecosystems captured within layers of permafrost contain not only biological material, but also environmental signatures. Pollen grains, plant fibers, fungal spores, and microscopic organisms reveal detailed stories about ancient climates. The presence of certain pollen species indicates temperatures, humidity levels, and seasonal changes at the time the soil froze. Microorganisms associated with plant roots show how vegetation responded to glacial cycles. These tiny fragments allow scientists to reconstruct entire habitats that no longer exist. Chemically preserved layers within permafrost also contain traces of ancient atmospheric composition. Carbon isotopes provide insight into the productivity of Ice Age vegetation, while nitrogen compounds reveal fertilization patterns of herbivore-dominated ecosystems. Studying these chemical signatures helps track the rise and fall of megafauna populations and understand how these animals influenced soil formation, nutrient cycling, and carbon storage. The thawing of permafrost exposes these biological and environmental archives with increasing intensity. Each summer reveals new remains, new microbes, and new chemical signatures. While some contribute to scientific understanding, others raise concerns about ecological instability. The loss of long frozen ground destabilizes terrain, turning once solid areas into wetlands, sinkholes, and collapsed ground known as thermokarst formations. These changing landscapes alter water flow, disrupt animal migration routes, and reshape vegetation zones. The biological processes unleashed by thawing permafrost influence local wildlife. Bacteria released from ancient layers interact with modern microbial communities, potentially altering nutrient cycles. Shifts in soil composition affect plant growth, which then influences herbivores and predators. The cascading effects can ripple through entire ecosystems, transforming food webs and altering habitat availability. These transitions occur at rates far exceeding previous natural changes, forcing species to adapt or migrate. Ancient materials preserved in frozen carcasses offer another dimension to these changes. Some frozen animals still contain partially intact blood, tissue fluids, and organ material. These substances carry biochemical compounds that provide clues about ancient pathogens. Proteins within blood samples reveal immune responses, allowing researchers to determine whether animals fought infections shortly before death. Patterns of inflammation in preserved tissue suggest interactions with parasites or bacteria. This information contributes to a growing understanding of how diseases shaped Pleistocene ecosystems and influenced species survival. Frozen remains also preserve evidence of environmental stressors. Bones with irregular growth patterns indicate nutritional shortages or harsh climate fluctuations. Lesions in tissue suggest injuries, infections, or predator attacks. These details offer a direct look at how Ice Age mammals experienced daily life. Their preserved bodies tell stories embedded in muscle fibers, bone structure, and even cellular damage. The presence of well-preserved hair in frozen animals provides insight into thermal regulation and seasonal adaptation. Microscopic studies of hair fibers reveal the density, length, and structural characteristics that allowed species like mammoths to endure extreme cold. Hair samples also contain traces of external contaminants, such as dust particles, plant fragments, and fungal spores. These contaminants act as environmental markers showing where the animals traveled and what habitats they frequented. In some cases, frozen specimens contain remnants of food between their teeth or within their digestive tracts. These remnants identify the plant species available during the animal's final moments. Analysis of stomach contents reveals seasonal variations in diet and helps reconstruct the nutritional ecology of extinct species. Such data also support studies on ancient climate shifts. indicating which plants flourished during cold intervals and which disappeared during warmer periods. While studying frozen animals and microbes provides valuable information, the process of excavation itself presents risks. Excavating permafrost requires careful control of temperature, airflow, and contamination. Tools used to cut through ice can dislodge ancient material, potentially releasing microbes into the air. Researchers wear protective equipment to minimize exposure, and excavation sites are monitored to assess environmental changes. Laboratories receiving samples follow strict protocols to ensure safety while analyzing biological material. The scientific need to understand ancient pathogens intersects with modern concerns about disease emergence. Even though viruses recovered so far from permafrost infect only single-celled organisms, the possibility of unknown viruses emerging from deeper layers attracts attention. Ancient pathogens may behave unpredictably when exposed to modern ecosystems. Their interactions with present-day organisms could differ vastly from their behavior thousands of years ago. These uncertainties encourage cautious research approaches. Despite these concerns, the study of frozen microbes contributes to advancements in medicine and biotechnology. Enzymes found in cold-adapted bacteria offer industrial and medical applications, functioning efficiently at low temperatures. Understanding how ancient microbes survive extreme cold may support innovations in cryopreservation, organ storage, and biotechnology. Knowledge of how microbes maintain structural stability under freezing conditions inspires new approaches to preserving biological material. Frozen fungi and spores likewise provide insights into resilience. Some spores recovered from permafrost exhibit thick protective layers, chemical defenses, and genetic pathways that enhance survival under stress. Studying these characteristics expands understanding of extremophile organisms and contributes to astrobiology research exploring the potential for life on icy planets and moons. Thawing permafrost also reveals clues about human activity in ancient landscapes. Artifacts preserved alongside animal remains include stone tools, carved objects, and remnants of early shelters. Human presence in these environments suggests that people adapted strategies to exploit frozen ground, navigate shifting terrain, and interact with large animals that dominated Ice Age ecosystems. When human-made tools appear near animal remains, they trace moments where people pursued prey, scavenged carcasses, or utilized natural ice formations for shelter and survival. Chemical signatures preserved in permafrost layers indicate the presence of ancient fires, pigments, or residues from plant processing. These traces provide information about human behaviors such as cooking, tool-making, and habitat modification. Studying this material reveals how early societies interacted with frozen landscapes and adapted to the challenges of extreme cold. The release of long-frozen methane and carbon dioxide from thawing permafrost also impacts global climate models. Ancient gases trapped beneath the ice contribute to warming trends as they escape into the atmosphere. Microbial activity accelerates these processes as organisms newly exposed to oxygen decompose organic matter and release additional greenhouse gases. The interactions between climate, biology, and permafrost form a complex web with consequences that extend far beyond the Arctic. The frozen world beneath the permafrost continues to reveal layers of biological information. Microbes, viruses, plants, animals, and chemical signatures preserve a record of ecosystems shaped by cold, time, and resilience. As thawing accelerates, more materials surface, expanding understanding of ancient environments and modern challenges. The melting of permafrost reveals more than isolated discoveries. It uncovers a silent timeline of planetary memory. Each thawed layer releases fragments of biological and geological history, preserved far beyond the reach of written records. As more ancient remains emerge from the ice, the distinction between past and present becomes increasingly blurred. Creatures that once shaped ecosystems return in forms so intact they seem displaced in time, carrying clues about environments that no longer exist. These revelations expand scientific understanding of life on Earth, tracing the interplay between climate, biology, and extinction across millennia. Permafrost behaves like a natural archive. Its layers form chronological bands reflecting shifts in temperature, precipitation, vegetation, and animal activity. As snow accumulated, compacted, and froze year after year, the ground captured everything that touched it. Pollen drifting in seasonal winds, the bodies of migrating animals, remnants of human activity, and microscopic organisms navigating complex soil networks. These layers preserved interactions between species and environments that long vanished from the surface world. When the ice begins to melt, it releases these fragments in unpredictable sequences. Bones reappear first, emerging through eroded soil or rising to the surface as the ground collapses. Soon after, more delicate materials surface: feathers, fur, plant fibers, and tissues that retain original textures. In some areas, thawing reveals ancient peatlands once covered by dense glacial vegetation. Their preserved roots and stems map ecosystems that supported the megafauna, whose bodies now lie exposed under the summer sun. The melting landscape reveals transitions that unfolded across thousands of years. Some frozen remains belong to animals that lived during relatively stable climates. Others originate from periods of rapid ecological change, when warming or cooling forced species to migrate or adapt. These differences appear in chemical signatures preserved within bones and tissues. Variations in carbon isotopes indicate shifts in vegetation. Nitrogen ratios reflect dietary stress or abundance. Collagen structure shows how environmental pressures influenced growth and resilience. As the ice retreats, these preserved records illuminate the factors influencing extinction. Several species found in permafrost declined long before humans expanded across northern landscapes. Climate fluctuations altered vegetation zones, forcing herbivores to adjust feeding patterns or relocate to unfamiliar territories. Predatory species responded to changes in prey availability, reshaping food webs and competition. By analyzing frozen remains, scientists reconstruct the complex relationships connecting environmental change and species survival. The chemical complexity of frozen tissue reveals subtle transitions that bones alone cannot capture. The presence of certain proteins reflects immune responses triggered by infections or environmental stress. Lipid structures indicate how animals processed nutrients during harsh winters. Microcracks in bone tissue show periods of dehydration or weakened health. Even the arrangement of hair follicles provides insight into thermal regulation and the severity of seasonal cold. Frozen environments preserved entire ecosystems down to microscopic levels. Samples of soil uncovered beneath thawing ground contain ancient fungal spores, bacterial colonies, and protozoa that once shaped nutrient cycles. These microorganisms interacted with plant roots, decomposed organic matter, and influenced soil chemistry. Their preservation offers clues to how ecosystems functioned during glacial periods. By comparing these ancient microbial communities with modern ones, scientists assess how climate change reshaped biological networks over time. Some of the most surprising discoveries involve frozen seeds and plant fragments capable of germinating under controlled conditions. In laboratory settings, seeds buried in permafrost for thousands of years sprouted new growth. These regenerated plants contain traits adapted to cold climates no longer found in modern species. Their DNA preserves genetic pathways lost through natural selection, as warmer climates replaced glacial ecosystems. These living remnants bridge evolutionary gaps and provide opportunities to study ancient plant physiology. The presence of human-made artifacts within thawing soils highlights another dimension of the frozen past. Stone tools, fragments of woven material, and traces of hearths appear near the remains of animals that once shared the environment with early humans. The permafrost preserves these objects with remarkable clarity, offering direct evidence of how people survived in harsh climates. These artifacts represent innovation and adaptation, demonstrating how early societies shaped their world and responded to environmental challenges. Some tools discovered in permafrost layers coincide with the remains of large mammals, suggesting that humans processed carcasses or used ice-stable ground for storage. Stone blades near frozen mammoths indicate butchering activities, while pigments found on animal hides hint at early forms of decoration or ritual use. The permafrost preserves these interactions in extraordinary detail, revealing how humans utilized natural cycles of freezing and thawing long before the concept of refrigeration existed. The discovery of ancient footprints preserved beneath melted ice captures fleeting moments from a distant past. Tracks belonging to children, adults, and animals appear where frozen mud once hardened rapidly. These prints show patterns of movement, revealing how people traveled, hunted, and played in environments vastly different from today's landscapes. Footprint depth, spacing, and direction provide clues to behavior, physical condition, and group dynamics. Permafrost thaw reshapes modern landscapes. As frozen ground warms, the soil becomes unstable, forming depressions, slumps, and thaw lakes. These physical changes influence water flow, vegetation growth, and habitat distribution. Rivers shift course, wetlands expand, and forests encroach on once frozen ground. These transformations disrupt habitats for modern species, forcing migration and altering the availability of resources. Animals that rely on stable ground, such as musk oxen and reindeer, face new challenges as traditional grazing areas change or disappear. The release of ancient carbon stored in permafrost contributes to warming trends. As organic matter thaws and decomposes, microbes consume it and emit carbon dioxide and methane. These greenhouse gases trap heat in the atmosphere, creating feedback loops that accelerate thaw. The biological processes occurring in thawing permafrost represent a significant factor in global climate models. Scientists analyze how microbial communities respond to warming temperatures to predict future emissions and refine climate projections. In addition to atmospheric implications, thawing permafrost exposes groundwater and soil systems to new chemical interactions. Nutrients locked in frozen sediment dissolve into meltwater, altering river chemistry and influencing aquatic ecosystems. Changes in soil structure affect root penetration, altering plant communities. These shifts cascade through food webs, impacting herbivores and predators alike. The transition from frozen to thawed ground transforms entire ecological networks, reshaping the balance of life in northern regions. The biological contents of permafrost reveal long-term ecological cycles. Some frozen plant fragments show scars from insect feeding, offering insight into ancient herbivore-insect interactions. Fossilized pollen suggests cycles of forest expansion and contraction during glacial and interglacial periods. These cycles shaped animal migration patterns and influenced human routes across the landscape. As permafrost thaws, the reappearance of these materials provides evidence for interpreting how ecosystems adapted to climate oscillations. Deep within permafrost layers lie the remnants of microbial ecosystems that thrived in oxygen-poor environments. These microbes metabolize nutrients differently from those found in today's soils. Their metabolic pathways offer clues about biochemical evolution and early life processes. By studying these ancient metabolic signatures, scientists explore how microbial communities responded to extreme cold, limited light, and minimal nutrient availability. These findings contribute to research in astrobiology and the search for life in frozen environments beyond Earth. The potential revival of ancient microorganisms from thawing permafrost raises biological concerns. While most bacteria recovered from ancient layers pose no threat to humans, their interactions with modern ecosystems remain unpredictable. Microbial communities compete for nutrients and influence soil chemistry. Introducing ancient microbes into contemporary environments may shift these dynamics in subtle ways. Studies aim to understand how such interactions influence decompositional processes, nutrient cycles, and plant growth. Beyond biological risks, thawing permafrost reveals structural instability in human-built environments. Buildings, roads, pipelines, and infrastructure constructed on frozen ground depend on its stability. As the permafrost warms, these structures experience uneven ground movement, requiring extensive repair or relocation. These socio-economic impacts intertwine with ecological transitions, illustrating how environmental processes influence human systems. The ice contains memories of landscapes that stretch back tens of thousands of years. It preserves ecological transitions that shaped the evolution of species, including humans. The more the ice retreats, the more it reveals evidence of migration, adaptation, and survival. Each thawed animal body, plant fragment, or microbial layer contributes to a broader understanding of Earth's past. These remnants of frozen time help reconstruct environments that existed long before written history, filling gaps in the story of life. The increasing pace of permafrost melt exposes both the richness of this archive and its vulnerability. Once exposed to air, preserved materials decompose rapidly. Organs collapse, tissues break down, and delicate structures disintegrate. The transition from frozen preservation to rapid decay challenges researchers, who must act quickly to document, extract, and study each discovery before it deteriorates. This race against time underscores the importance of scientific collaboration and ongoing fieldwork in Arctic regions. The deeper the thaw penetrates, the older the materials it reveals. Layers formed during early glacial periods emerge after remaining undisturbed for tens of thousands of years. These ancient strata contain organisms that predate many modern species. Their presence captures moments from epochs when Earth's climate, atmosphere, and biosphere differed radically from today. The biological and chemical signatures preserved in these layers expand understanding of how life endured and evolved during extreme conditions. Frozen bodies recovered from permafrost offer a unique glimpse into the genetic diversity of extinct species. DNA extracted from their tissues outlines evolutionary pathways that shaped megafauna biology. Genetic sequences indicate how populations migrated, adapted, and interacted with other species. These data help clarify why some species survived, while others disappeared. They highlight the complex interplay between environment, physiology, and genetic resilience. As thawing continues, permafrost transitions from a stable archive to an active agent in environmental change. Microbial respiration increases, releasing greenhouse gases. Waterlogged soils accelerate decomposition. Plant communities shift in response to new nutrient patterns. These processes transform the Arctic from a region defined by frozen stability to one characterized by rapid ecological flux. Understanding these shifts requires integrating insights from biology, geology, chemistry, and climate science. Within this dynamic landscape, ancient remains emerge not as isolated discoveries, but as components of a larger story. They reflect the resilience of life under extreme conditions and reveal how ecosystems respond to prolonged cold. Their preservation demonstrates the capacity of ice to protect organic matter far beyond its expected lifespan. The information encoded within these remains enriches scientific understanding of ecosystems shaped by forces far greater than individual species. The frozen world continues to open as temperatures rise. Each newly exposed animal, plant, or microbe contributes to a record written in layers of ice and soil. These remains expand knowledge of how ancient species lived, interacted, and adapted. They reveal environmental patterns that shaped the evolution of life. As the thaw accelerates, the archive changes, losing some records as others emerge. Don't forget to subscribe. It really supports the channel and helps us bring you more great stories.